Okay, so Assalamualaikum warahmatullahi wabarakatuh and a very good day everyone. So welcome to the webinar Scientist Talk Malaysian COVID-19 Vaccine Research and Development. So I'm Hamida, Dr. Hamida Idris from Biology Department, Faculty of Science and Mathematics, UPSI, and I will be moderating our webinar session for today. So the webinar is conducted as the course assignment for my two subjects, SBT 3013 Biotechnology and also SBB 3033 Principles in Microbiology as preparation for their Sulam project in Hello Microbes 6, and also one subject SBT 3012 Principles in Biotechnology under Dr. Nuhaida Kamarudin, where in this webinar the topic of COVID-19 vaccine R&D in Malaysia will be discussed to get some insights and information from the person that involved in this area firsthand. And we would like to know on what actually happened behind your BSL3 lab door. Right. So before we begin the session, I would like to share some housekeeping notes. The first one, today's session is being recorded to be available for viewing afterwards. And if you have any questions to our questions, please use the chat feature in this WebEx webinar, which will be addressed after the talk. And for students of SBB 3033, SBT 3013, and SBT 3012, do not forget to write in your class name and metric number in the question. Right, so attendance link will be given throughout the session, so please fill them in and feel free to write your comments and feedbacks. So now without further ado, we will turn the time over to our speaker for today, Dr. Siti Nurzawani binti Rosli, who is a research officer in Infectious Disease Research Center, IDRC, Institute for Medical Research, IMR, and she's the one who is responsible for the development of inactivated COVID-19 vaccine in Malaysia. So prior to this, Dr. Zawani worked as an education and research support staff and postdoctoral researcher in Hiroshima University, Japan for 5 years after the completion of her PhD at the same university, Hiroshima University, in 2013. She obtained her Master of Science and Bachelor of Science from University of Malaya in year 2010 and 2003. But not only excellent in science field, our speaker today is also blessed with various talents, including voiceover, narrator, MC, and perhaps singing. So now, moving along to our session, please welcome Dr. Zawani, who will be speaking to us on Malaysian COVID-19 vaccine R&D. Over to you, Dr. Zawani.
Thank you, Dr. Hamida. Assalamualaikum and good afternoon, everyone. Can you guys hear me? Is it okay with the audio?
Yes, okay.
All right. So everyone knows that prevention of disease by vaccination is one of the most significant achievements in medical research history. It has become a key to the cost-effective reduction of mortality, improvement of life expectancy, and economic growth. One way of meeting vaccine inequity is to enable developing countries like Malaysia to produce their own vaccine. Assalamualaikum and good afternoon, everyone. Thank you my name is Siti Nurzawani Rosli. People usually call me Wani or Dr. Nini. I'm a research officer from Institute for Medical Research, IMR. IMR is now under the National Institute of Health, NIH, located in City Alang. I'm also a wife, a mother, and also an old friend to Dr. Hamida Idris. Thank you, Dr. Hamida, for the opportunity to talk with your students today on the topic development of COVID-19 vaccine in Malaysia. Okay, before we go deeper into our topic today, I would like to bring all of you here in NIH, National Institute of Health, located in City Alam. Dr. Hamida, is it this talk, is it like two ways? Is it possible for me to communicate with the audience?
Yes, but I mute the attendees, but they can answer in the chat. Is that okay with you? Or you want everybody to
I
be
cannot
unmute?
see the I cannot see the but it's okay. I will ask the question and answer it myself for now. All right.
Or you want
Anyway,
them to you want me to unmute them? Which one would
is
you
it
prefer?
okay? Okay, let me try
No
for 5
problem.
minutes. If it's like not not nice, then we mute everyone back. All right. So any of you ever, okay,
It's
listening to
very
us,
cold,
right?
Okay, kau confirm lah
Oh,
record.
students, please mute your microphone.
Everyone, please mute your microphone. I promise you my voice is very soothing. Hi, everyone.
Okay, Wani. Or would you prefer everybody to be mute?
Alright lah, mute lah. Macam-macam bunyi ada tu.
Alright, so
Alright,
maybe they will
thank you, Dr.
respond.
Hamidah.
They will respond in the
Okay,
chat.
tak apalah.
I think.
Yeah, I know, you know me, Vida. I always talk to myself, so it's okay. And then, okay, first of all, I would like to bring all of you here. NIH, National Institute of Health, is very beautiful complex just next to City Alam Highway. And look at that, it looks like nice lah. It's white and gray, it's very, very suci macam tu. It consists of 10 blocks. And there's over there IHM, IHR, all of the research institute and the Minister of Health are placed under one roof and one management. Next slide, please. Okay, this is the organization chart of National Institute of Health. So NIH is the research body under MOH. There are 6 research institutes in here with one management, and we merged together in 2019, and we are known since then as the National Institutes of Health. And NIH is under the supervision of the Deputy Director General. I'm sure everyone know our Director General, DG, our famous DG Nur Hisham. And the under DG Hisham, there's a few deputies. And for a Deputy Director General for Research and Technical Support, it's also another Dr. Hisham, Dr. Hisham Shah. So NIH is responsible in conducting health research that cover initial planning, prioritizing research areas according to the needs. So for example, for the past 2 years, our priorities went to pandemic COVID-19. So all of the research institute and the MOH are focusing to tackle the pandemic. Each institute under NIH responsible to conduct research based on their niche. For example, if it's about biomedical research, the portfolio will be taken up by Institute for Medical Research, IMR. If it's about clinical research, the research will be in charge by the ICR and so on and so forth. Other than conducting research, we also provide training and consultation for the ministries and other agencies as well. So if you need some consultation or require training or information or advices in related to Malaysia healthcare in Malaysia, you are welcome to come and see us. Next slide, please. All right, I'm going into Institute for Medical Research because that's where I'm currently placed. I'm from Institute for Medical Research. Has anyone of you ever heard about IMR? Dr. Hamida, are you aware? Are you since when did you know about IMR? When you were a student, are you biasa dengar IMR tak?
Yes, during undergraduate.
How about your students? Are they really
Okay,
aware about the
somebody
existence of IMR?
Somebody answered. Inek just said it sounds like brain scan.
What brain scan? All right.
MRI.
Okay. Yeah. Basically, we are like the MRI of the research in biomedical Malaysia lah. All right. So IMR, it's been here for 100 of years already. It was established 100 of years already. Before this, we were located next to HKL in Jalan Pahang. So the building next to the building next to HKL too, already 100 of years over and it's still there and looking like 100 of years, 100 years old already, right? So it was established by the British IMR in 1900, and during that time it was known as a pathological institute. The aim during that time was to promote the health status of the local population and to explore on the infectious disease tropical medicine. And since over the years, the function of IMR has been expanded to other areas as well, such as cancer research, environmental health research, allergy and immunology research, the non-communicable disease very related to Malaysian, cardiovascular, diabetes, and nutrition, herbal medicine research, which is also very important to our community, which because I think you also realize that our community is still much dependent on herbal medicine. Specialized diagnostic center, they are handling all the rare disease diagnostic. Medical research resource center, where they are in charge of all these expensive instruments, and the biggest of all is the infectious disease research center, where I'm from. And infectious disease research center, if there's any outbreak, including the recent pandemic, we are the one who are handling the diagnostic and the, making policies for going through the pandemic. All right, and next slide, please. All right, and then any questions in regards to NIHIMR before we go to our main topic? Belum masuk topik lagi, I tahu Ustaz. Natamida, I should proceed.
There seems yes, there seems no question in the chat. So for the participants, if there's any question, just pop in your question in the chat, and I'll read it up for Dr. Zawani to answer. Thank you. Okay, go ahead,
Alright,
Rani.
then let's proceed to our main topic, development of COVID-19 vaccine in Malaysia. Alright, the recent outbreak COVID-19 is pandemic. Pandemic is when the outbreak spread all over the world. I mean, there's like all over the world lah. But for the endemic, it's also all over the world, but we already learned how to live with it and tolerate the pathogen, like we are trying to do with COVID-19 right now. All right, and I think one of the biggest threats to civilization is the pandemic. From history, we learned that disease outbreak that spread all over the continents has brought many powerful nations to their knees, including what happened in Rome. And at the start of 21st century alone, the world has dealt with a few pandemics already that happened very close to each other, such as H1N1 and Zika outbreak. And the biggest and most alarming pandemic in these modern days, definitely the COVID-19. Slide, please. All right, I just want to share with you the timeline of COVID-19, the progress. Okay, this COVID-19 started in Wuhan on 31st December. Wuhan Municipal Health Commission in China reported a cluster of cases of pneumonia in Wuhan, and then they identified novel coronavirus. And then in January 2020, WHO set up the IMST, Incident Management Support Team. This is part of the emergency footing for dealing with outbreak. And then WHO reported on their social media that a cluster of pneumonia cases with no death was reported in Wuhan. In the same month also, China publicly shared their genetic sequence of SARS-CoV-2. And this genetic sequence has been used by all the countries all over the world to prepare should the outbreak came to their countries. And in the next month, in February 2020, WHO announced official name for the virus and the disease it caused. The disease is called coronavirus disease 2019, COVID-19, and the virus is named severe acute respiratory syndrome coronavirus 2, SARS-CoV-2. And in the same month also, the WHO China Joint Mission was formed, which includes experts from Canada, Germany, Japan, Nigeria, Republic of Korea, Russia, Singapore, and US CDC NIH to go to Wuhan to understand and observe the situation and report back to WHO. And following that, in March 2020, WHO declares COVID-19 as a pandemic. And the first case of COVID-19 in Malaysia was reported on January 25th, 2020 by our former Menteri Kesihatan, Datuk Dr. Zulkifli Ahmad. Okay, and then this slide, I want to show just a very, very short summary of COVID-19 history in Malaysia since the first case since the emerging of the first case on January 25th, when from the three Chinese nationals that traveled from Wuhan and tried to enter Malaysia from Singapore. And then the event, attended by 16,000 people, eventually triggered the wave of COVID-19 infections across the country in February 2020. And this was and form was dubbed by what was dubbed as the Tabli cluster. What happened was the attendees who contracted the virus during the event brought it back home to their respective states, resulting in a spike in infections nationwide. And then in March 2020, we experienced the first MCO, Movement Control Order. This is because in March 2020 is still new, the outbreak is still new, COVID-19. So we don't know much about the virus. We don't know much about the disease. There was no definite medication to treat to treat the disease, and there's definitely no vaccine. So the only way we can put an outbreak under control is through isolation. Therefore, government imposed a movement control order to their citizens, and the border was closed, and and there's also interstate travel ban. And this measure proved to work because in July 2020, for the first time, we managed to record zero local transmission. All right, and then but the third wave started in September through the Sabah state election, and over at the peninsula, we also seen workplace and dormitories of foreign migrant workers clusters. If you can remember the story about Top Glove, they made headlines after Malaysian government announced that it opened 19 investigations paper into their subsidiaries over offences involving their dormitories. And then following that, there's in January 2020, we had another MCO proclaimed by our king. But during this MCO 2.0, what we call it, the restriction was more lenient, and maybe that's why it's hard. And in MCO 2.0, the cases are not very much under control. And then in February 2021, our Malaysia's COVID-19 immunization program kickstart. And I was among the earliest to get vaccinated, and I was at my third trimester pregnancy during that time. The guideline during that time was only allowed for the healthy adult to get COVID-19 vaccination. But I made my case, and I managed to get my jab. And then after that, we have another last MCO 3.0, maybe around early May 2021. I was about to give birth, and I gave birth during this MCO 3.0. And then the rest of history, so many histories. I think if I want to tell everything, COVID-19 Malaysia timeline, maybe we'll finish 1 hour only about that. Okay, I think we should move on to the next slide. The lesson that we can get from COVID-19 pandemic, there's so many lessons, but one lesson that I want to highlight for today is on the vaccination. I think because of this pandemic, so many people, the public especially, can now understand the importance of vaccination. And other than that, for us as scientists, this is also kind of an opportunity, a door for us, because finally we can make the stakeholders and the government understand why it's important to have our own vaccine research, our own capacity to make local vaccines. Right? All right. Now we go to our topic. What is vaccine? According to the WHO 2018, it is a biological preparation that improves immunity to a particular disease. It typically contains an agent that resembles a disease-causing microorganisms. It often made from weakened or killed form of the microbe, its toxins, or one of its surface proteins, or a vaccine could be the agent that stimulates the body's immune system to recognize the agent as foreign, destroy it, and remember it, so that the immune system can more easily recognize and destroy any of these microbes that it later encounters. So how the vaccine works, this is a very simplified form of it. So when the vaccine gets introduced into our body in a safe amount, this vaccine is the antigen that will help the immune system to recognize them as hostile, and then the body will develop antibodies towards it for future infections. So how is a vaccine beneficial? Of course, it is for the health gains. Vaccine has been proven over and over time that it reduces mortality. I think you can see after our vaccine, COVID-19 vaccine program, we can see the death, COVID-19 death in Malaysia reduced significantly, and not just Malaysia, all over the world. It also benefit in healthcare cost saving because it's a simple concept. Vaccine is much cheaper than your hospital bill. Okay, it's much cheaper to get vaccinated than to get treated in hospital, pay for your all the services in hospital and your medication. And then vaccine is also beneficial in care in a way it's in care related productivity gains, because there will be no need for parents to care for their sick children, thereby saving productivity work time for the parents. It also beneficial in outcome related productivity gains, because it will increase productivity because vaccination improves cognitive functions, physical strength, and people benefit from individuals being vaccinated. This is what we can get through herd immunities. Because some people cannot receive vaccines, there's health problems, and what we can do is for us who can receive vaccines, we make ourselves stronger for these people. All right, and through less antimicrobial resistance, what it means is when you get vaccinated, you are stronger from the inside. So it will be less likely for you to get antibiotics, to be treated with antibiotics. And if you don't take antibiotics frequently, that means there will be less likely for you to develop antimicrobial resistance. I'm sure all of you understand, because I was made to understand that you are from microbiology students, is it, Dr. Hamida?
Yes, there are three courses, microbiology and biotechnology, but those taking biotechnology have taken microbiology before. So yeah.
All right, so I'm sure you can understand that. But if you don't understand, you can ask Dr. Hamida. All right, next slide. Oh. All right, we go to the history of vaccine. I'm going to touch a little bit on this. So the practice of immunization dated back hundreds of years ago. For example, Buddhist monk drank snake venom to confer immunity to snake bite. And they also practice what we call as variolation. Variolation is smearing of a skin tear with cowpox to confer immunity to smallpox. This was practiced in 17th century in China. And anyway, much earlier before that, variolation has been practiced in Ottoman Empire. It's a Muslim country. All right, this is based on a letter to a friend written by Lady Mary Wortley Montagu. Lady Mary is a wife of a wife of British ambassador to Turkey during that time. So in her letter, she told a story that she witnessed variolation in Constantinople in 1717. And then following year, her son was variolated in Turkey, and her daughter received variolation in England in 1721. The procedure was initially met with much resistance, so much so that the first experimental variolation in England was carried out on condemned prisoners who they were promised freedom if they survived, but they did. And nevertheless, the procedure was not without danger because during that time, the variolators, English variolators, devised the techniques that's not very safe. All right, and then later, the variolation techniques was replaced by much safer vaccination described by Dr. Edward Jenner. Dr. Edward Jenner, this picture showed Dr. Edward Jenner, who is considered as the founder of vaccinology in the West in 1796. He was considered so after he inoculated a 13-year-old boy with vaccinia virus, the cowpox virus, and then that boy demonstrated immunity to smallpox. And in 1798, the first smallpox vaccines was developed. This illustration shows the story about the success of smallpox vaccine found by Edward Jenner. In his book, he wrote that cowpox is a disease transmitted from horse to cows, and it appears on the nipples of cows in the form of irregular pustules. Eventually, these pustules will degenerate into vaginal ulcers. As a result, milkmaids, due to their proximity to the cows, ended up contracting the illness as well, and they suffered from inflamed spots on different parts of their hands, which eventually led to tumors in their armpits. Symptoms including shivering, overheating, quickened pulse, vomiting, and pain throughout the limbs. However miserable these symptoms may sound, Edward Jenner observed that the contraction of this illness was not so unfortunate after all. These milkmaids, after recovering, developed antibodies, not only made them immune towards cowpox, but to smallpox as well. And thus, cowpox is a much milder version of smallpox, and Edward Jenner concluded that to prevent smallpox, one must purposely infect themselves with cowpox. This procedure of immunization through inoculation was introduced and named as vaccination, derived from the Latin word for cow, vacca. This portrait shows the Dr. Edward Jenner, who lived from 1749 to 1823. And the next, this picture show the pamphlet published in 1806. This pamphlet in today's day is also known as journal. Published in 1806 by a group of physicians in Philadelphia. The title, Smallpox Destroys, Vaccination Saves the Lives of Thousands. This is in support of Dr. Jenner's work and the vaccination procedure. This pamphlet was published to urge the medical establishments and the public to adopt vaccination. All right, I'm going to explain a bit on the vaccine immune response. It's quite complex, but I'll try to make it simple for you. Dr. Hamida, have they covered this topic in their courses?
In microbiology course, yes.
Yeah. Is it like deep or on the surface?
On the surface, just a
Alright.
brief mechanism.
Okay, so I'm going to refresh your memory lah, refresh your knowledge a bit. So the vaccines, there's antigens, right? So the antigens, so the vaccine antigen when it gets introduced into your body, the APC, antigen presenting some cells that has a lot of arms. Okay, why it gets so many arms? Because their duty is to fetch these antigens, and then they will process it and present it to the T helper cells. That's why they are called antigen presenting cells. The name already so clear like that. And then the antigen presenting cells will present this antigen to either CD4 positive T cells or CD8 positive T cells. Okay, let's go to the to CD4 positive T cells first. So when the CD4 positive T cell capture these antigens, it will get activated. Before that, they are called naive T cells. Now, after they gets activated by the antigen, they will be called effector T cells. So these activated T cells can secrete two types of cytokines. Cytokines number two first. We go see at the bottom. If they secrete Th2 cytokine, T helper 2 cytokines, these cytokines will in turn activate B cells. When the B cells gets activated, it will make these B cells to either proliferate to make more memory B cells, or they can produce the neutralizing antibodies, which is very important if you're talking about vaccines. So these neutralizing antibodies, what it function for? It's to neutralize your pathogen. So this is very important now for a vaccine to be therapeutic in mechanism. Okay, and then another way CD4 positive T cells can exert its immune response is by secreting Th1 cytokines. If you can see in the middle, Th1 cytokines, what Th1 cytokines do is it will activate CD8 positive T cells. CD8 positive T cells is also known as a cytotoxic T cells. Why is it called cytotoxic T cell? In comparison to the CD4 positive T cell, which is called T helper cells because it only helps around. But for the cytotoxic T cell, it it's there to do the real job. Okay, so the real job, that means that it goes straight to the pathogen and kill it. So when it gets activated by the Th1 cytokines, it will either proliferate to make memory T cells, or when it gets activated, it can straight away kill the infected cell. There's another way CD8 positive T cell can get activated other than through Th1 cytokines is from APC straight away. APC can activate CD8 positive T cells straight by presenting the antigen through MHC1 to CD8 positive T cell. And then when it gets activated, it will become cytotoxic T cells. All right. Any question and for this one, all of you? Or we proceed?
So far, there's no question in the chat. Dr. Wani, just
All
proceed.
right. Am I going to cepat or is okay?
I think you're doing just fine.
All right. Okay, now we go to the type of vaccines. Okay, typically, many vaccine candidates will be evaluated before any are found to be both safe and effective. For example, of all the vaccines that are studied in the lab and laboratory animals, roughly 7 out of every 100 will be considered good enough to move into clinical trials in humans. So only 7% that is developed in the lab can go through clinical trials. So that means that it feels somewhere in the middle. And of the vaccines that do make it to the clinical trials, this 7%, just 1 in 5 is successful. So 7% and 1 in 5 of the 7% is successful in the clinical trials. Therefore, having lots of different vaccines in development increases the chances that there will be one or more successful vaccines that will be shown to be safe and efficacious for the intended populations. Therefore, we need more vaccine study in Malaysia. So far, there's only we can count in 5 fingers. So we have to start more vaccine research so that the chances for us to have one successful vaccine will be increasing. All right. Okay. There are 3 main approaches to making vaccine. Their differences lie in whether they use whole virus or bacterium, just the parts of the germs that trigger the immune system, or we only use the genetic material that provide the instruction for making specific proteins and not the whole virus. So we go back again. We can make vaccine using the whole organism or only parts of the organism that can trigger the immune system, or we only use the genetic material. All right. Oh, okay. Okay, let's go to the first approach, the whole microbe approach. For the whole microbe approach, there's three platforms in the development and in fact in use today. Number one is inactivated vaccine. Um, for this platform, we make a vaccine using the whole organism, and we inactivate it or kill it using chemical, heat, or radiation. This approach uses technology that's been proven to work. Okay, we have so many inactivated vaccines in the market, and most of the vaccines that are in use today, many of them are using inactivated platform. So example of these vaccines, in example of inactivated vaccine is polio and flu vaccine. For this vaccine, it is easily to be manufactured and grow in a large scale. However, for this vaccine, the making of the vaccine will require special laboratory facility to grow the virus or bacteria, and it will require long production time because we will be depending on the propagation rate of the organisms that we're gonna use. I mentioned before, for this vaccine, we're gonna need a special laboratory, right? Especially for those of risk group 3 organisms, if we are going to make vaccines vaccines using the risk group 3 organisms, we are going to need the BSL-3 laboratory. Okay, examples, the recent COVID-19. In order for us to develop this kind of vaccine, one thing we must have is the biosafety level 3. Okay, and then for this vaccine, usually it will require booster dose. And the next whole microbe approach is the live attenuated vaccine. This kind of vaccine uses a living but weakened version of the virus. Example of this vaccine is measles, mumps and rubella MMR vaccine, chickenpox and shingles vaccine. For live attenuated vaccine, it uses similar technology like the inactivated vaccine. It's just that for inactivated vaccine, we completely inactivate the microbe. But the live attenuated vaccine, we just make it weak, and we have to make sure that they cannot replicate in our body. And therefore, vaccines like this may not be suitable for people with compromised immune system because we are scared that we don't know, even the weakened organism, what happened in the compromised for those in the body of compromised immune system. All right, and the next one is the viral vector vaccine. It um, uses safe virus to deliver specific subparts of the germ of interest so that it will trigger an immune response without causing a disease. For example, okay, for the viral vector vaccine, as the name implies, vector, that means we use the different another virus to deliver the genetic material of the pathogen that we want to fight. Okay, so for example, the latest COVID-19 vaccine lah, when we use AstraZeneca use this technology, and also this Sputnik V, where they use adenovirus as a body. But inside the body of the adenovirus, we insert the genetic material for COVID-19. And then when this virus replicate, it will also replicate together the protein of COVID-19. In this case, most of the vaccines of COVID-19, they use spike protein. The genetic material that's been inserted inside this adenovirus is also the sequence that code for spike protein. Okay, and then the adenovirus won't harm us but it will carry together the COVID-19, the spike protein of COVID-19, and our body will recognize the spike protein as a pathogen, and then will develop antibody towards it. All right. Example of this viral vector vaccine is the one that I just mentioned to you, the COVID-19 vaccine, and also Ebola vaccine. All right, the next platform is the subunit vaccine. For this vaccine, it only uses the very specific part, what we call a subunit of a virus or a bacteria the immune system needs to recognize. So it doesn't contain the whole microbe. So for this picture, they only show the spike. So I'm using another COVID-19 example also. So if a COVID-19 vaccine is to use subunit approach, they only need to put the spike protein inside your body, and then our body will recognize it, and then incite immune response towards it. All right. So the subunits may be proteins or sugars, and most of the vaccines on the childhood schedule are subunit vaccine. For example, whooping cough, tetanus, diphtheria, and meningococcal meningitis. The last type of vaccines is the genetic approach. This gets very famous recently during the pandemic. I'm sure all of you have heard about Pfizer, BioNTech, Moderna, Johnson, all of them. No. Pfizer, BioNTech, and Moderna, both of them are using the mRNA platform, right? So for this one, how can I simplify? Okay, for the mRNA virus, if mRNA vaccine, they use I'm using the Pfizer, BioNTech, and Moderna as an example here. So the mRNA vaccine, you can synthesize the mRNA for COVID-19, our COVID-19 vaccine, mRNA COVID-19 vaccine. There's only instruction to make spike protein. So this mRNA is packed into the what we call it, a lipopolysaccharide. There's special recipe for it, and then we put into our body, and then this vaccine will use our cellular machinery to synthesize proteins according to what mRNA sequence inserted into the body. And then the proteins that are synthesized later will be recognized by our body as a foreign, and then our body will will respond. Our immune system will respond and produce antibody towards it. All right. So DNA, sorry. So this nucleic acid approach is a new way of developing vaccine. Actually, this is very latest technology for vaccine. And before COVID-19 pandemic, none had yet been through the full approval process for use in human. But for DNA vaccines, it's been there's a few DNA vaccine for cancers, but still under human trials. But because of the pandemic, research in this area has progressed very fast, and mRNA vaccines for COVID-19 are getting emergency use authorization, which means now we can use them. Okay, but we still don't know what is the effect long term. But so far we are good. All right. Okay, this slide just shows the summary of platforms for COVID-19 vaccine. We have whole virus vaccine, subunit vaccines, viral vector vaccines, and DNA and RNA vaccine. Okay, this slide shows the traditional vaccine development pathway. A vaccine usually will take about around 12 to 15 years until it finally obtains the license for public use. But for COVID-19 vaccine, its development was expedited in so many ways. So you can see the bottom one, a typical vaccine development is linear. As you can see, straight line. Each step is completed before going to the next. But with COVID-19 vaccine, the top one, Some steps were overlapped. As you can see, the green one, during the clinical trials, phase 2 clinical trials started before phase 1 even finished, but all steps are completed in its entirety. And then the next one for the manufacturing, as you can see, it's overlapped with the clinical trials. So manufacturing has started even before clinical trials was finished. Okay, that's how COVID-19 vaccine development was expedited. Okay, then the COVID-19 vaccine has been added into list of vaccine preventable diseases. To date, there are 8 or more, I think by today it's more, COVID-19 vaccines granted with emergency use listing by WHO and FDA. So we have here the most popular in Malaysia is Pfizer-BioNTech, Sinovac, and AstraZeneca. Okay, this slide show simplify and summarize how vaccines for COVID-19 been developed so fast. So number one, funding. The funding was easier because everyone comes together. Governments and funding bodies join forces to remove financial obstacles. They themselves override the system they put. All right, and then priority and collaboration. Scientists, doctors, ethics approval boards, manufacturers, and regulatory agencies have all come together to work harder and faster. For example, our regulatory agencies and PRA, there was no guidelines into getting some biologic vaccines or medicine that is not complete in their clinical trials. But because of this COVID-19, they have to come up with new guidelines to bring these biologics and let Malaysian people receive it. Okay, and the science advances in vaccine technology that are built on the back of many years of research. For example, the mRNA vaccine, before this is just in the lab. Okay, no one have ever used mRNA vaccine, but mRNA vaccine has been under development since 20 years ago, and this COVID-19 pandemic is seen as the opportunity to finally get these technologies in use. And then for the manufacturer, large-scale manufacturing of the vaccine occurred in parallel with the clinical trial to scale up production quickly. So all these big manufacturers put other things aside. They are all came together to manufacture COVID-19 vaccine first before they proceed with others. And then volunteers before these clinical trials, like the clinical trials, the operator have to recruit the volunteers. But for COVID-19 vaccine, volunteers came to the clinical trial center, volunteering themselves for the trial, so that everyone, so that they are part of the saving the world apa namanya, mission for the COVID-19. All right. And then, all right, we are going to highlight aspect of vaccine. Dr. Hamida, is there any questions from the crowd? I'm taking 5
Okay,
seconds.
Dr. Zamani. Okay, so let me read this. The first question from Wong Yuhui from biotechnology class. I have a question with the part of failure in developing vaccine, right? As there's only one
with the what?
the part of failure,
Part of failure.
part kegagalan in the developing vaccine. Okay, so as there only 1 in 5 of 7% is successful developed,
Yes.
was there a clear issue or reason prove the failure along developing process?
Oh,
And
developing
was the
1 in 5. Oh, sorry. Yet, Dr. Hamida.
okay, so the other part, and was the failure of vaccine adapt to host, especially in embedded into DNA chain, be one of the reason?
Okay, the 1 in 5 is failed in clinical trial. So when you are failing in clinical trial, usually is in regards to the percentage of usually, okay, for the clinical trial, they have to fulfill the safety and immunogenicity. So there are percentage, or there are some skill they need to fulfill. For example, for WHO, you have to pass 70%. Different vaccines has different guidelines. For COVID-19 recently, you have to 50% efficacy. So if you don't achieve that 50% efficacy, your vaccine will be failed in clinical trial. And have you ever heard about dengue vaccine that's failed in the Philippines? They also failed in clinical trial. And another reason, there's so many reasons you can fail in clinical trial when you have death cases, so many deaths, and there's AFI, unexplainable adverse side effects. So what you find in many of it, that's about clinical trial, not in the technical anymore. So the 7% too, yeah, so the 7% that success in R&D. All right. So, but of course, um 7% success, at 7% success, the other 93% fail too, does not mean you fail 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 like 100% fail. That means that you are giving the other team information of things that they shouldn't do. Okay, so just continue with your research and don't forget to share even you fail, so that other people wouldn't repeat the same thing. And then about the DNA just now, Dr. Hamida, I actually didn't get the question. You mean that the DNA vaccine?
Let me just read the question again.
Okay.
Was the failure of vaccine adapt to host, especially in embedded into DNA chain, be one of
Mm-mm.
the reason failure?
If it's at first cannot, meaning that the DNA vaccine embed into host DNA.
Maybe I will allow Wong Yi to communicate with
All right.
Dr. Zawani.
Yeah, that would be
Alright.
best.
Yes. Let me find. Let me unmute. I'm going to unmute you, Wong Yi. You can ask question to Dr. Zawani. I can't unmute this participant. All right, I can't unmute Wong Yi. I'm so
All
sorry,
right,
Dr.
it's
Zawani.
okay. Okay, maybe she or she can communicate with me later. Okay, I think we should proceed because I have half. All right,
Dr.
so
Zawani, wait.
yes,
I have
yes.
another question. Are you
Oh,
okay
what
with
is
another
it?
one question from Muhammad Fahim?
Fine, but I
Right.
don't know whether you are okay with it.
That's fine. One question here,
All
right?
right.
If Malaysia finish in developing our own vaccine, do we have to
Mm-hmm.
take the vaccine that are produced by Malaysia even after 3 doses of vaccine? He's asking, are we going to take Malaysian vaccine after this?
Of course, you have to. Because I think if we have our own product, why don't we use it? But for COVID-19 at the moment, I think it's gonna be like flu. It's gonna be we have to, because it's gonna be forever around us. So to tolerate living with COVID-19, we have to be prepared. I mean, ourselves have to be prepared. So how we want to be prepared living with something notorious like that is like get ourselves protected by taking the vaccine. So I think it's gonna be a routine vaccine. But I don't know. Let's hope for miracle. Something. I mean, maybe the virus will mutate into something that is less harmful. Inshallah. There's two way of it. It's either miracle or we have to be prepared all the time until we die.
All right, thank
Okay.
you. But to add in further curiosity, Dr. Zoni, is it okay? Let's say for
Yes.
the first and second and third
Uh-uh.
doses we took
Mm-mm.
AZ or Pfizer
Mm-mm.
or whatever, and the fourth one
Mm-mm.
we're gonna
Mm-mm.
take IMR vaccine. Will that be okay?
Mm-mm.
Will that be like suitable for the body or something?
Theoretically, it won't be a problem. But all of these things, whatever question that we have in our mind, The theory we can answer, but we don't know what happened if in real setting. So that's why there's preclinical trial and clinical trial. So theoretically, I think it should be okay because our immune system respond to the same thing, the spike protein. So but as a scientist, all of you also a scientist, we have to prove what we think in our mind in the lab setting. All right. So it's still our responsibility in IMR to study that. Okay, Dr.
Thank
Hamida?
you. Okay,
Okay.
thank you so much to
All
you.
right.
Okay,
So
just
let's
proceed.
move on with our next topic, halal aspects of vaccines. And before that, disclaimer, I am not expert in this halal aspect of vaccine lah. So everything that I'm going to say on halal aspect vaccine is like on in my opinion. All right, not I'm not going to impose whatever I believe in on all of you, but this is what I have in mind on halal aspect of vaccine. Okay. So in Islam, Muslims are required to follow Sharia law. So the Holy Quran states in Surah An-Nahl, ayah 114 to 115, therefore, O believers, eat of the lawful and good things that Allah has provided for you, and be grateful for His favors if it's true that you only worship Him. Indeed, Allah has forbidden you that which dies of itself, so Allah forbade us to eat that which dies of itself, blood, and the flesh of swine, also any flesh consecrated to something other than in the name of Allah. So four things. But whoever is compelled by necessity to eat any of this, not intending to sin or transgress, will find Allah most forgiving, most merciful. So in Darura, Allah will be most forgiving and most merciful. So this passage explains why Muslims abstain from using haram material or consuming porcelain products and derivatives. Okay, and so in Malaysia, our muftis have agreed that COVID-19 vaccine is halal and obligatory. Our muftis agreed of this matter is based on two factors. Number one, istihala, and number two, istihla. What is istihala? It refers to alteration of physico-chemical nature to change a non-acceptable haram product to an acceptable halal form. Alright, while meanwhile, istihala, mixing, can convert an unclean substance into one that is clean. So istihala refers to mixing. Sorry, why is it my? Istihala refers to mixing of a substance with another until it is dissolved, causing loss of properties even though the substance still exists. Thus, an unclean product can be mixed with a more dominant clean product to vanquish the unclean characteristics. This concept is adapted from the hadith, which explains the characteristic of tukola. This concept is adapted from the hadith, which explains the characteristic of tukola of water. Prophet, the Prophet was asked about the status of stagnant water being licked by reptiles and wild animals, whether the water is still clean. Then the Prophet said, if the water exceeds 2 kola, then it does not become unclean. Another hadith explains, if the water exceeds 2 kola and is then mixed with the unclean, it does not become unclean as long as there is no change in its smell and taste. However, the concept of istihalah and istihla are not universally accepted by all ulama and authorization councils which determine halal status. All right. But for Malaysia, our muftis opted this concept to permit the use of COVID-19 vaccination recently. And some of this suggested that if the concept of istihala is to be applied on a vaccine or any biologics, this vaccine must be studied in the laboratory to check whether there are traces of this haram element still remains in the vaccine. But this paper showed that the summary of animal or human origin excipient traces in the vaccines that we use for COVID-19 And they concluded that there was no trace of animal or human origin excipients at all in this product. So inshallah, our COVID-19 is halal. All right, if it's according to the istihalah and istihla concept. Okay, and Jakim has also came out with a long information on vaccine and immunization in their website. I'd like to highlight some of their points here to simplify things for our discussion today. Number one, it is indeed forbidden for us to use haram resources in medication. Number two, however, prevention and seeking medication is wajib or obligatory for a Muslim. Therefore, in the case when there is no other options or resources, medication from haram resources will be permissible. In Darura, it will be permissible. So I hope this will help build confidence in us to get vaccinated and to help convince others to get vaccinated as well. All right, and then we shall go to our next topic, development of the first inactivated COVID-19 in Malaysia. How it is all started? This just 50% of our team. All these vaccine experts in Malaysia are in here from UPM and also from IMR. So how it is all started? Malaysia is not equipped with neither vaccine development nor vaccine production facility. But since vaccine production is not the main focus of our country prior to COVID-19 pandemic, this field remains as one of the least explored. So COVID-19 pandemic has made us realize the importance of having our own capacity to produce our own vaccine. So realizing the need to finally strive towards our local vaccine capacity and taking the opportunity of having our BSL-3 at our disposal, IMR came together with vaccine experts from UPM, University Putra Malaysia, aiming to develop a locally made COVID-19 vaccine in April 2020. During that time, everyone was locked in at home. So we came together and then we discussed the possibility to start our own vaccine production because at that time the procurement also we faced so many problems for the procurement of our vaccine as well because we have to rely on this vaccine manufacturer outside. We have to beg them to give us something. All right, so from there our stakeholders and our ministers realized that this is the time we have to start, and therefore we came together. UPM came to us and approached us with the idea, and then with our BSL-3 there, new, beautiful, and not utilized, and then we have not utilized, so we say yes, let's start the project together. All right. So, but I mean, we have so many platforms like we just discussed before, but the only option for us during that time is to proceed with the inactivated COVID-19, inactivated platform. That's because this platform needs the least technological advances and least complex for us to work with. Therefore, we think that by doing inactivated vaccine, it will increase the chance for the vaccines to be successfully produced. Okay, next, this slide shows the overview vaccine strategies. I want to show to you different vaccine strategies and why at last we opted for the inactivated vaccine strategies. I'm going to take a second break. That's us.
So one of the Teletubbies is Dr. Zawani.
Yes, I was heavily pregnant when I'm doing the first part of the what we call the project, and then during the defense, we have to defense to our panel of the top management why we want to proceed. I was also pregnant. I was very sick, but it's okay. Finally, we succeeded to get the money and everything. Okay, so I couldn't there are so many pictures of us in our BSL-3, but pula when the time I want to use it, I couldn't find it because there's too many maybe. All right, so lastly, we chose the inactivated vaccine platform because of the simplicity and its safety profile. We know that it's going to be safe, it's going to work, and we have the facility, and that's why we proceed with inactivated vaccine. Okay, so mostly due to the readiness of the resources to develop this vaccine platform. So opportunity provided by we have the SL3 and we have the isolate. Because during that time, I think all of you, maybe if you go to the media, read the news during the pandemic, I think IMR has been in the media every day during the pandemic. We have been leading the country into dealing with the pandemic. We have to come up with the manual. We have to handle the diagnostics. We have to come up with the answers from the media, the answer from you guys, and then we've been working 24 hours, I think, during this pandemic and all of this. All right, and then therefore, because we are the main handlers of the diagnostic COVID-19 diagnostic in Malaysia during that time, and also the reference laboratory, we have so many isolates, or so many COVID-19 isolates at our disposal. So we can use this isolate to develop our vaccine. So, as you have known, the inactivated COVID-19 vaccine consists of SARS-CoV-2 particles that have been grown in culture and then killed. And then killed, right? So we must have lots of virus to make this vaccine to be used by our population. Right? So we must be able to grow this virus in a large amount. All right. Okay. And next, I'm going to tell a bit on the history of inactivated vaccine. Inactivated vaccine for human dated back in 1930s. I have to mention for human because inactivated vaccine also have been is widely used for animal. Our poultry also vaccinated. If there's no vaccine for our poultry, maybe we don't have a healthy chicken and healthy cow for us to eat. All right. So the experience of inactivated vaccine used by Salk to develop the inactivated polio vaccine in 1954, and following that, Dr. Jonas Salk vaccine from Dr. Jonas Salk has helped in getting rid of polio once and for all. And in 1986, the inactivated hepatitis A vaccine was developed by Provost and co-workers, and all these vaccines use chemical inactivation. There are also other inactivated vaccines currently in use, such as Japanese encephalitis and tick-borne encephalitis virus. Okay, as you can see in my slide here, manufacturing process of inactivated vaccine, I'm going to go very brief on it. So we have virus here, and then we have so in the isolate in our lab, so maybe inside there maybe got like 50 virus particles. So how can it be enough? So the first step, we have to increase the number of virus. We must propagate the virus in mammalian cell lines. Okay, once we have to culture the virus, increase the number of virus, and then when we have enough number, we inactivate the virus. That means that we kill the virus by chemicals, and then we must make sure the virus is pure, clean from the cell debris by purification and impurity removal process, and then we put the vaccine in a nice bottle like that, and then it is now ready for distribution. Okay, and then I'm going to share with you a little bit on the development of inactivated COVID-19 vaccine by IMR. We started this project in 2021. The project was in plan since April 2020, but we must prepare the proposal. We must defend the proposal and ask for money, and we have to fail once and twice and three times, and only we pass to get the fund. And then finally, we get the money in January 2021. And then the first step of vaccine development is to identify the vaccine candidate, the suitable. Like I told you, we have so many vaccine virus isolate in our archive. So which one is the best one? Because there's no more turning back. Once we already grow one or two or three candidates and fail, so we must have a special characteristics to determine, and we have to have the reason why we choose this one isolate. All right. So a suitable vaccine candidate was identified from the archive based on a few characteristics. So in our case, there's a special characteristics that um uh, we defined. All right, and then after we already identify this isolate is the best one, I believe this would be this way inside a strong immune response, and then we propagate the virus upscaling process. All right, but we have to not have one candidate lah, because during the propagation there might be the possibility this candidate cannot be propagated. It cannot give us the title that we require to make a good vaccine. All right, so after you manage to upskill the virus, you have to ensure the genome of this vaccine is stable using whole genome sequencing. So after you are ensure, oh, this propagation does not change the virus, because you know virus is very vulnerable to mutation, because one or two or three mutation is like different person already. All right. So we have to make sure while we are propagating, the virus does not change from the original isolate. Okay, so after you make sure, or after you propagating millions of copies and it does not change, so that's the best vaccine candidate. All right, and then after you have done that, now your vaccine is already involved. It is ready for inactivation. In our case, we inactivate our vaccine using chemical. All of this process must be done in BSL-3, because right now, as you can see, one or two COVID-19 particle also can infect you and make you get sick, right? So right now we have like millions of copies of SARS-CoV-2 in our lab. So it is very dangerous. So everything must be done in BSL-3. Okay, and then after we get our vaccine seed, it is now ready to be formulated. Formulated. So in formulation means that you can add any other ingredients in your vaccine to make your vaccine special or much stronger, or up to you. How do you want your vaccine to be? And then after formulation, you put in your vaccine in a nice bottle with label and with name. Okay, nama yang sedap. And then now your vaccine is ready to be used in preclinical trial. You must test the safety and efficacy of your vaccine in mice or lab animals. Okay, right now we are at preclinical trial, 60% of it, and then we hope that we can finish by December 2022. Okay, I'm going to touch a bit on the selection of vaccine candidate. Our vaccine candidate, we chose our vaccine candidate based on clinical representation, how severe is the clinical representation by the patient, and the ability for the isolate to be propagated. That means that the isolate must be able to grow in a large scale in a short amount of time, and the genomic stability. We mean the genome of the virus must be stable. It's not easily mutated. All right. So I would like to show with you the clusters that we have during the second outbreak and during that time, and then we get our isolate from these clusters. All right, and then this is how we chose our vaccine candidate, how we analyze the clinical representation and the genome and the genome sequence of the virus. And then this is how we ensure the genomic stability of the vaccine candidate. All right, so you have to make sure the what we call it, the mutation acquired during the several passaging does not change the virus so much. It's still within the what we call it the same family. All right. So how to know that? We have to uh, construct phylogenetic tree to see all of the passage does not cause huge changes, does not in the genetic sequence, the genomic sequence. All right. Okay, and then after we are ready with that, we propagate our virus and we check the genomic stability, and then it's ready for inactivation. This is the picture show the inactivation processes which happen 2 days in a row. So we have to be prepared. We have to come visit the lab until 24 hours. Okay. And so the inactivation was done using chemical in our laboratory, BSL3 laboratory. And then this is how we check for we confirm the inactivation when we try to, when we inoculate our vaccine inside the cell culture, there was no cytopathic effect seen on the cell. That means that the virus has been inactivated fully. And another way we confirm the inactivation is through PCR. Okay. And then I would like to touch a bit on vaccine formulation. In a vaccine, what do we have in a vaccine? We must have the antigen, that is the active ingredients. And a vaccine could also have preservative, stabilizers, surfactants, residuals, diluent, and adjuvant. So for preservative, what preservative does is to prevent vaccine from become contaminated, and stabilizers to make the vaccine stable. So therefore, stable, how to make a vaccine stable so that there is no chemical reaction happen in the vaccine bottle. And surfactants, as the name implies, to make sure that the ingredients inside the vaccine does not clump together, and then they must be residual, especially during the manufacturing. All right, residual usually does not affect anything because it usually presents in part per million residues, and diluent, that's usually the water. Okay, some vaccines came in the like lyophilized form. So you have to dilute the vaccine with water. Usually it's water lah, the what we call it, double distilled water. And then our vaccine contains the addition of adjuvant. All right. So adjuvant in our formulation is uh, uh, is um is to keep the vaccine at sorry, to incite stronger immune response. How adjuvant incite stronger immune response is by keeping the vaccine at the injection site for a little longer, so local immune system can get stimulated longer time. Okay, and okay. Next one, and now okay, now our vaccines are ready in a bottle. You can see the picture, that picture. Okay, you can see this is our formulation and bottling of IMR vaccine candidate. We did this in the GMP facility in UPM. That's Prof. Hair, one of our important collaborator. He's holding our vaccine that's been inactivated. And right now it is ready for bottling. There's a bottling ready for mixing, formulated with adjuvant. And then this is the D picture shows the bottling process, and then the E picture is how our vaccines is now ready, and then the C picture, as you can see, that's how as our vaccine ready for preclinical trial. And now our vaccine is ready for preclinical trial. Okay, preclinical study. Preclinical study is to test new biologics such as drug and vaccine in animal. This is very important before clinical trials in human can be done. Okay, data from this study is required by our regulatory agency, is the NPRA, National Pharmaceutical Regulatory Agency. So many people don't know this. Our regulatory agency in Malaysia is the NPRA. Okay. So preclinical study, we have to do this. and we have to generate information on the immune response by the vaccine, effective doses, and we have to make sure that it's safe. So we have to come up with the safety data. Okay, and then after all of this, only then we can move to clinical trial. Okay, this is some of the pictures from our preclinical study that we conducted in Veterinary Research Institute. Okay, before I forgot, our inactivated COVID-19 vaccine project is collaboration project between three institutes, that is IMR, UPM, and VRI. The VRI stands for Veterinary Research Institute. So all of these institutes from different ministry, but we put our differences aside and then came together to make sure our first inactivated COVID-19 project from Malaysia successful. Okay, and then the aim, every our preclinical study is still underway. We still have so many months after we can finish, but this is how the scene, the A1, we always have so many things with us when we want to go there. And the B picture is our virologists and our pathologists in action. This is the most beautiful situation of us. And then C, me and my loyal assistant Rahma is still with me, who's with me also. And the D is how we immunize the mice, and then the E picture is the mice that we use, special humanized mice that we use for the preclinical trial. Okay, and then next, I would like to touch a bit on the challenge vaccine production and manufacturing challenges in Malaysia. The first challenge that we face is oh is I have to say the fund. We came up with the plan to develop this project. For this project is in 2020, and we only secured the money in 2021. So I guess you can imagine the first huge challenge is the fund. So we tried two times, one from MOSTI, and then the next one is from MOH, and finally MOH agreed to invest a few million, not a few million, 1 million plus into our research for the duration of 2 years, only for the until preclinical stage. And then the next big challenge it was to identify the facility to perform safety and efficacy study for vaccine in animals. In IMR, we have biosafety laboratory level 3, 4 in vitro study, but we don't have one for animal. So after scouting around, finally I found one in VRI. And that's when VRI joined us because they have the facility and expert to handle animal BSL-3. VRI located in Ipoh, so we have to visit Ipoh every week during the preclinical trial. Okay, and then the next challenge is preparation for clinical trials. So for clinical trials, you have to produce your vaccine in this special certified facility, but we don't have that because we never produce vaccine on our own. Therefore, what for is the facility before? So right now, without the facility, we cannot move on from proof of concept phase. So another milestone, and another challenge that we have to deal with and still not solved until now is to get a facility that is certified to make vaccines that is safe, that will allow us to go beyond proof of concept. And another challenge is preparation for manufacturing. But it's okay right now. We have started engaging with potential manufacturer for the vaccines, and because of this, the R&D field and manufacturing field has now seen each other and building bridge that before this is not exist. Okay. And then other than that, we have also must work with the, it's okay. I think prior to COVID-19 pandemic, human vaccine production was not a popular research topic in Malaysia. So the pandemic has tested our country in many areas, especially our healthcare system in their ability to handle crisis at such a huge magnitude. So the recent pandemic has made us realize the need to have our own vaccine production and manufacturing capacity for at least to be self-sufficient. So, therefore, experts from different institutions have come together and come up with a formula to establish a vaccine production and manufacturing ecosystem in Malaysia. This is how we want to move forward. So, the effort to move forward in this vaccine research and production in Malaysia has been undertaken through the National Vaccine Development Roadmap, a roadmap that will be overseen by the Ministry of Health, the Ministry of Science, Technology and Innovation mostly. Um, and this roadmap has identified the areas that need to be completed or upgraded so that Malaysia can finally become a vaccine manufacturing country and could be the main hub for halal vaccine provider. There will be so much potential in Malaysia should we go into vaccine production because I think there is no central hub for halal vaccine. So I think it's good that we tried to in this field halal vaccine production. Okay, I think that is all. Thank you very much. Is it on time? Right. Oh. Okay, Dr. Hamida. I hope I don't exceed the time given.
Right. Thank you so much to Dr. Zawani. We have lots of questions
Okay.
and all all of them are very long ones. So I
Okay.
think if we
Okay.
try to address each and every single one of the question, it might take till like 8 p.m. or something.
All right.
So
So maybe
I
I'll
would like
take
to make a
three questions and then the rest maybe I'll answer in a writing form.
Yes, I
Is it
was
okay?
about to make a request from you. Is either we do one recorded version of Q&A, I mean like answering questions from you to be
Oh,
shared
boleh,
with the
boleh.
students.
Boleh. Yes, yes.
Ah,
That would be
would
great.
I know
Sebab
you prefer
I malas nak
talking than
tulis. I
writing, isn't
jawab
it?
terus.
I know. You
Yeah.
boleh tulis, tayo pemalas kan?
Yeah.
So
Tak apa.
maybe
It's
one
okay.
session of, like asking question and you explain, the
Right.
answer and everything. And I'll
Yes.
share them with the student. But
All right.
there's one simple question
Right.
that I would like to ask you, taken from the attendance form.
All right.
Okay, so the statement is, I'm scared of vaccine. 3 doses are
Okay.
already enough.
Mm-hmm.
So, comment on that, Dr. Zawani?
It's okay as long as you um uh just amalkan SOP. I think it should be fine. But um because this thing we cannot see, right? I don't know how whether this if we are alone who practice all this SOP but the other person around us does not. So the only way we can equip ourselves with with our own guns is I mean our own what we call it protection is by vaccine. So all your fears, so you have to overcome by your own.
Is the like take the look
You
of the benefit of
yeah,
the
yeah, you have to
vaccine.
take the benefit. Yes, I that one I cannot help because that one it has to be from you. It's I think that means that she or he already got the 3 doses.
Yes.
The,
Maybe I
so that
think
means that that means that the vaccine has done nothing to him or her lah. So it's okay to get the fourth one then.
Maybe that's the common question among the public is that how long are we going to, really rely on this vaccine? How many more jabs? And when will it stop? So, yeah.
That I told you before, number one, we have to depend on a miracle. We have to pray that the virus might mutate to something that is less harmful. And so number two right now is we have to find a vaccine that works with one doses only. So I think as a scientist, we must keep studying. Maybe someone will be successful in making a vaccine that only require one dose, like what happened to our children. There are just longer, I mean, lifelong immunity, a vaccine that will incite lifelong immunity in your body. So don't be the one that's hoping, be the one who made it, who make it.
All right. Okay, thank you so much. It's already 4 o'clock and the questions are still coming. It's okay for the students. You can just add up the question. I'll copy everything and, bring and discuss with Dr. Zawani about it. All right. So I think we need to end the session and it's a very, very good one. Thank you so much to my beloved friend, Dr. Zawani. We have known each other since, I don't know, teenage
Tahu
years.
lah berapa tahun. Don't tell the numbers. They will know that we are so old.
Yeah, I'm not old.
Yeah, me too. That's why we just do 2
Okay.
years, Dr. Hamida.
Yes. Okay. So thank you so much to Dr. Zani. We are glad that you could be
All
here
right.
today to share your
Alright.
experience, knowledge on COVID-19 vaccine R&D. So for the students, I hope this session will convince your worry about the safety and efficacy of COVID-19 vaccine because as what have been presented by Dr. Zawani, it's not that easy that we can just mix like making orange juice to produce one vaccine. Right, so there are lots of ethics, SOP, and regulations to be fulfilled, right? And plus the critical trials and everything, right? So that's why the challenges, there are lots of challenges faced by them, the person in charge of this COVID-19 pandemic. So on our end, we might just say, why this, why that, why using so much money, why taking so long? But they have their challenges too. So the best thing that we can do is, like doa, that everything will be smooth and the virus will, I don't know, finally mutate and fade away or something. So, and from the session, I hope it answered the vaccine ingredient, right? About the vaccine ingredient and also the mechanism, especially for SBB 3033 students, where you need to present this to the kids during the Hello Microbe 6 program. So please make use of all the informations and, utilize them and share with the community, right? So I think above all that I can conclude the real important thing is the motivation and determination that what makes this possible and turn this Malaysian vaccination effort into a reality. And thank you so much. We are so lucky to have Dr. Zawani and the person behind this started from the proposal development, defense, like what you did during your FYP where you have to present your proposal and the panel's asking you about question. That's what she did. And she failed also. And she have to repeat it again and again, right, Dr. Zawin, a few times till she get the funding. So life is like that, all right? So yes, thank you so much. I hope Dr. Zawani, there might be another session or something because there's a comment. About the comment, this session is being conducted in English. I'm so sorry. We do it in English this time because in course SBB 3033, we have international students, so we need to cater their needs. But if there's request, we might have this in Malay version if, yes, if you would like, anybody would like to invite Dr. Zawani, we can contact her or contact me and let her know about it, right? So thank you so much for everyone. Don't forget to fill in your attendance form. We are now at the end of our webinar, Scientists Talk Malaysian COVID-19 R&D. So Dr. Nurhaida and me, Dr. Hamidah binti Idris, would like to thank our speaker. Million thanks to Dr. Siti Zawani and our audience for today for your participation. Thank you so much, everyone. See you again next time, inshallah. Thank you.
Thank you, everyone. Good luck. Assalamualaikum.
Salam. Good luck for the assignment, Dr. Wani.
Bye. Assalamualaikum.
Bye. Thank you. I'll catch up with you after this, okay?